I never know. I'm a little bit
Did I mark you here?
Yes.
You just left for a minute?
Yeah.
Alright, so these are water molecules. Now, the first thing you need to know is why the water molecule is the way it is. We say we say the molecule is a polar molecule. Y'all ever heard that?
Yep.
Polar? Chaz, are you with me up here?
I am.
We gotta do AP bio. Sorry.
AP.
Thanks. So we say water molecule is a polar molecule. Why do we say that?
Because
The electrons are shared unequally.
Very nice. We say it's polar because the oxygen side is negative and the hydrogen side is positive. And you need to understand why that is. They're going to test you on that. Why is the oxygen side negative? Well, go ahead, Thomas.
Because it has the electrons closer to it. It holds it closer.
Yes. So these electrons here are shared electrons. They're shared between the oxygen and the hydrogen to create the covalent bond. Remember, covalent bond is sharing electrons? So you can fill your outer level. Are y'all with me? I need y'all to pay attention here. So the electrons are shared between the hydrogen and oxygen, but they're pulled more closely to the oxygen. So it creates, since electrons are negative, the oxygen side is negative. And the hydrogen side is positive. So whenever you have water molecules that are near each other, like they won't approach each other like this. Because both of those are negatively charged, it'll flip it around. So they're like that. And here comes another one just coming up wanting to be friends. And nope, it has to turn around that way. You see what I'm saying? And so all the water molecules, they'll take on this kind of regular arrangement where they kind of look like this. Now they'll only arrange themselves nicely like that if they're cold. And you'll form what we call hydrogen bonds between the molecules. Do you all remember that the property of water that ice expands when it freezes? Y'all know that?
Yeah.
Y'all put beers in the fridge, in the freezer, right? When your parents are out of town, right? I know about. And it blows up the bottom. And you have this huge mess. And you forget about it. And then mom comes home. And you get busted. So what happened was, the water molecules, when they're warm, they move around quickly, you see? And these hydrogen bonds won't hold them in place. But when you freeze water, they kind of slow down and they take on this regular arrangement and they leave spaces between them. See, if they were moving faster, they'd fill those spaces. But since they slow down, they leave spaces between them and the water expands. It expands by about 10%. Its volume gets about 10% greater. Well, that's enough to break a bottle. It's actually enough to break a rock. It's the strongest erosive force on the planet. Erosion. Have you all ever heard of erosion? You know, if you build a road and you just leave it there for a few years, there'll be cracks all over it. You know why? Because the water gets down in a little crack, and then it freezes when the weather gets cold, and it expands, and it breaks the road. That's why they always got to fix the road. Roads are way better in places where it never freezes. You know who has the best roads in America?
Florida.
Georgia.
Actually, yeah, Florida.
We're number one, baby. And part of the reason is because we're south.
South Carolina.
Why wouldn't Florida be better? Because it's even further south and even warmer than us? Because Florida's lame. We spend more money on our roads in Florida. I-95, baby. 3 lanes. All right, we good? Did you understand water and kind of how it works? They'll ask you about water. Sharing electrons. It's a polar covalent bond. There's all sorts of properties of water. You know, these hydrogen bonds, even if it's warm and these things are moving around, the hydrogen bonds still exert force. And so that's why water forms droplets. What's some other properties of water? Do we remember?
Yeah. Surface tension.
Cohesion, they stick together and form droplets. Surface tension. So the surface of water, if you look, if these water molecules are all on the surface, there's no hydrogen bonds pulling up from the air. This is air up here. There's no hydrogen bonds pulling up, but there's hydrogen bonds pulling down. So all the water on the surface gets pulled down. And what it does, it kind of, if you pull all these down, you kind of pack them together. And the surface of the water is harder, really, than underneath. Adhesion is sticking to another surface.
Capillary action.
So like water will stick to glass, or will stick to this board. If I flick some water, it would just stick there probably. And that's adhesion. So adhesion is sticking to something else. Cohesion is sticking to yourself. But it is because of hydrogen bonding. Yeah.
Capillary action.
Yeah, capillary action is if you take a glass tube and you stick it on some water, the water will kind of run up the glass tube because it sticks to the sides. And you get what's called a meniscus. Do you remember that from chemistry?
Mm-hmm.
Kind of goes down, it's stuck to the sides, so it dips down in the middle a little bit. So that's because of adhesion.
What's the importance of like the high specific heat?
Okay, so because of these hydrogen bonds, it's like, with specific heat, I used to imagine that these bonds are like little springs that attach the molecules to one another. And do you know that molecules are always moving? Unless they're at absolute zero, molecules are always moving. This is my molecule dance that I do at Ziggy's. And if you slow molecules down, they don't move as fast. Specific heat is water will really hold heat well because all the water molecules are like connected to one another. So imagine we got a whole bunch of water molecules connected to one another and you get them all vibrating real fast. You with me, Chav?
Yeah.
They're all vibrating real fast, but imagine they're connected to one another with little strings. If you want to cool that water down, you got to stop all those molecules from vibrating. And they're all connected to one another. Don't you think that would be difficult? As soon as you slow this molecule down, if you let go of it, it'll speed back up because it's connected to all these other molecules. So in other words, it's hard to cool down water, and it's hard to warm up water because all these connections between the molecules.
So it has a high specific heat?
It has a high specific heat, meaning it can hold a lot of heat in it.
And it's a low what? A low... Is there...
A low.
Something low. Like a low...
I don't know what it has
I
itself.
might be wrong in that.
I'm not sure. But anyway, it can hold a lot of heat. You know, it takes a long time for water to cool down. You go out at night after a summer day, the water's still warm, you know, even though the air's cooled off. So it really holds heat well. Okay, let's move on from water. Organic molecules. Remember these? There's 4 types of organic molecules. What's the first type? These things.
Monosaccharides.
What?
Monosaccharides.
That's a subdivision.
Carbohydrates.
Carbohydrates.
Unbelievable.
No, that's good. You are right. One type of carbohydrate is a monosaccharide. It's just a single ring structure. Carbohydrates just have carbon and hydrogen and oxygen. You see the oxygens, you see some hydrogens. Where are the carbons?
Where the beds are.
Everywhere where you see a point is a carbon. You assume that they're there. And so, if a single ring carbohydrate is called a monosaccharide, can you give me some examples of monosaccharides? Fructose. Fructose.
Sucrose.
No, not sucrose. There's one other one, galactose.
I could forget galactose.
It's easy to forget galactose. Glucose looks like this. It's a hexagon. Fructose looks like this. It's a pentagon. They actually have the same structure though. Both of these are C6H12O6. Even though there's only 5 rings, because there's a carbon down here and a carbon up there that make it 6 carbons. So they're both C6H12O6. Now, what if you take a glucose and a fructose and you hook them together? What do you have? Not a monosaccharide. You got a disaccharide. Can you give me some examples of disaccharides? Sucrose is one. Lactose is the other one we hear a lot about. Ooh, somebody said maltose. Who said that? Kame. Do it, Kame. So these are all disaccharides. Now let me show you that the curriculum wants you to know how these things form bonds. Oh, here we go. Here's a glucose molecule. Here's another glucose molecule. Now, we have 2 types of reactions that you need to know. You have what's called a condensation reaction. It's also known as dehydration synthesis. Are y'all with me, Matt? Are you with me? Oh, good job, man. How about you, Julie? You with me back there? All right, here we go. Here's 2 glucoses coming together. How are we going to join them together? We take an H off this one and an OH off this one. All right, flip it over. What do we have there?
H2O.
H2O, that's water. And these things are bonded together. That's dehydration synthesis. That's how big molecules get bonded together. We call a single unit a monomer, and we call many units together a polymer. So a monomer is a single unit. Mono means one. You know Monopoly? You ever? Did you know you're looking at the national 1986 Monopoly champion?
Of course.
I don't know that.
Are you being serious?
Thought you might want to know.
Wait.
Can you win
Well,
Monopoly?
Number 1, mono.
You can win Monopoly?
Well, there's a big Monopoly tournament I got in.
Where?
Geneva, Wisconsin.
I know it's a joke, you see?
Cereal.
Your cat.
I actually don't
Took 3 days.
I don't know.
Geneva Convention? Or like
You won?
Yeah, the Geneva Convention, Gen Con. You ever heard of that?
What lots did you get?
What?
What lots did you get?
Oh, I got them all. I had them all by the end of it.
We start with
Here was what I did to win this thing.
trains.
Whenever somebody was going broke, I would buy their properties from them at ridiculously, at really high values. Way more than you'd think it's worth. And I would almost bankrupt myself in buying everyone's properties. But I had all these properties which I had to mortgage. I had to, when you turn them over, do y'all know how to play Monopoly?
Yes.
I had all these properties that I couldn't even use. I was almost broke, but I had all the properties. And I just keep going around, pass and go, and every time I open a few of these properties up, and pretty soon I won. And every time I did that strategy every time, like 4 or 5 straight games. And it was a big tournament, like a bracket tournament. And no one else ever did it.
How many people?
I won every time.
Was it 1v1? Like the
No, it was like 6 of us in
Wow.
each game.
How many people were there in the whole tournament?
What year was this?
Well, like 100 or something.
1986.
150. Yeah, 1986, Gen Con. They're not going to have it on there, but I don't think we had the internet back then. But it was fun.
It's a real thing.
I'm gonna find it.
You can find it. I mean, mine might be on there, but if you find it, let me know.
Willis just said.
All right. So, enough about me.
Talk about background.
So here we go. These two come together. Water comes off. H2O, right?
Yeah.
That's dehydration synthesis. How do you break up a large molecule?
Put in water.
What's the name of the reaction?
Hydrolysis.
Hydrolysis. Here comes water. It comes in. It attaches. The molecule breaks apart. Just add water. That's why you soak your dishes. The water gets into the molecules and breaks them apart. Now you know why you soak your dishes. Do y'all soak your dishes? You just throw them there. Let the maid get them? Is that? Okay.
I don't know.
Now, so what if we hook together many of these monosaccharides in just a long chain? One after another, after another, after another, after another. We can make a very large molecule, can't we? There's almost no end to the size of the molecule we can make. What is this big thing called?
Polysaccharide.
Polysaccharide. Can you give me some examples of polysaccharides?
Starch.
Starch.
Cellulose.
Cellulose. This is the most common molecule on the planet other than water. It makes up plant cell walls,
What about nitrogen?
which is what if you have a piece of wood, it's almost all cellulose. What's that? What else?
What about nitrogen?
Wait, what's the?
No, that's not a molecule. That's an atom.
Yeah, that.
But isn't it two nitrogens bonded together?
Oh, nitrogen gas.
Yeah.
I think there's more cellulose by weight, because that's just the atmosphere. The atmosphere is pretty thin. It's a good question. You can look it up.
No, not me.
It might be that cellulose is the most common organic molecule. It could be. Look it up. Starch, cellulose, what else? Glycogen. That's liver sugar. And glycogen is important in our studies of biology. We'll get to liver sugar second semester. Diabetes, you ever heard of that? Involved there. What does Reddit say?
I would.
You still working?
Still working.
Starch, cellulose, glycogen, those are good polysaccharides. I thought there was another one.
You just
Well,
look
there's
at
probably
it.
lots of them.
There's a lot of them, but another one that's common.
Wait,
Starch, cellulose, glycogen. Chitin.
Meryl just said that. She's like,
You did, Meryl?
Yeah.
Meryl.
There you go, Meryl.
Speak up, that's good. Chitin is the cell walls of fungi. And it's also what like your fingernails and animal hooves and stuff are made of. It's the exoskeleton of crabs. Chitin's a big one in biology.
We just part crab when you think about it.
Are we good with carbohydrates?
Yes.
Y'all good with carbohydrates?
Yeah.
All right. Since we're good with carbohydrates, we'll go on to lipids. What's a lipid? Fats. So lipids are made of these things. What do we call these things?
Fatty acid chains.
Nice. Fatty acid chains. And it's usually have several fatty acid chains hooked to one of these things. You know what this is?
Wait, starts with G.
Glycerol.
It's glycerol. This is how I heard it.
Cammy.
Cammy, you're on fire.
Getting there.
Glycerol hooked to fatty acid chains. It's nice. I like hooking it to 3 of them. That's kind of my thing. There you go.
We all got that.
You know what we call that? That's a triglyceride. That's the fat that's under your skin. That'll keep you warm, store food for you. That was big in the caveman days. People liked it. Everybody, the healthiest ones were real fat. They were going to survive the winter. Skinny people, all of us, probably
So,
would have been cast out of the clan. Figured we were diseased.
How big would they get?
Oh, they get as big as they could. They didn't live very long, but so glycerol with fatty acid chains, that's a fat. Now, sometimes the fatty acid chains aren't straight line. You know what we call them when it's all single bonds? We say it's saturated. Saturated fats, you ever heard of those?
Yeah.
They're usually not as good for you. They'll clog up your arteries and such.
Is that the lipids?
Butter,
Yeah.
saturated fat. Anything that's solid at room temperature. You know why it's solid at room temperature? Because see how tightly these can pack together? You could take another glycerol here and put it next to it and just pack them in tight. So it forms a solid fat. Blubber is all saturated fat. What's that? So the bent. So if you throw in a bent side chain,
Unsaturated?
that's an unsaturated fat. And it's because there's a double bond in the structure. And that double bond takes up it can't have as many hydrogens, so we say it's not saturated with hydrogens. There's some hydrogens missing because of the double bond. So it's an unsaturated fat.
Are they liquid?
Unsaturated fats have a kink to them, and that causes them not to be able, if you put an unsaturated fat over here too, the kinks make it so they can't get as close together. So they're liquid at room temperature, like oils. Oils are unsaturated. And they're usually a little bit better for you. Eating olive oil is better for you than butter if you're going to cook. Now, if you throw, let's take this one off. Watch this. I'm gonna take this one off. There's only two, and I'm gonna put.
Oh, I know it.
I'm gonna put a phosphate group right here, and a little nitrate group right there, and now you know what we have?
Phospholipid.
That's a phospholipid. These are lipids. Lipids are the fats. And this is a phospholipid. And that's a very common molecule. It makes up your cell membrane. Millions of these things get together and line up. Some will line up on top. Do y'all remember some line up on bottom?
Yeah, phospholipid bilayer.
Yeah, to form the phospholipid bilayer.
I asked myself.
Mr. Willis?
Yes.
What are the parts of this phospholipid?
Parts of a phospholipid?
Yeah.
We got two fatty acid chains. One's saturated, one's unsaturated. We got the glycerol. We got a phosphate. And we got this nitrate group. It's called a choline group usually.
It's like an R group?
Actually, every one of them is not like this. They can differ. Sometimes there's different fatty acid chains. Sometimes you don't have the choline. Sometimes you have a different group up here.
Is the choline
There's a whole bunch of actually different types of lipids in the membrane, but this is the basic type.
Is the choline just considered an R group? Is an R group?
The choline, yeah, an R group, when we say something is an R group, that's just kind of a general term for the rest of the molecule.
Okay.
So, it's not just R groups on lipids. Amino acids have R groups. R just is like a variable meaning other stuff.
Okay.
We good on lipids? Lipids are fun.
Yeah.
Okay, then, maybe the most important
Steroid.
is the amino acids. What'd you say?
I said steroids.
Steroids?
Or you don't use them. It's not just natural.
This is natural, yeah. I don't have steroid magnets up here.
Yeah.
I have steroid magnets, I just don't have them here. Steroids is another type of lipid, which is important. We'll learn more about them when we do hormones. Proteins are made up of what? What are these little things?
Amino acids.
They're chains of amino acids. What is the structure of an amino acid? You should know this.
It's a chain.
It's an amine group.
Oh, not that
I put a carbon in the middle. Can you figure out the rest on your own? Could you write it down in your own little notes?
Honestly, no.
There's an amino group, there's an amino group. And a carboxyl group.
Now, remember, it's called an amino acid.
It's an amino group.
So, you put an amino group on one side. There's your amino group. They might test you if you know that that's an amino group. There's an acid group on the other side. So here's your amino group. Here's your acid group over here. What's your acid group?
C.
C.
And then double bond O.
O.
OH.
OH.
Oh my god, I'm a genius. Okay.
That's your acid group.
And the R group.
It's also called a carboxyl group.
Wait, so
Are you with me over here, Chan?
I'm with you.
Mr.
Can
Willis,
it see this?
I can.
Okay.
the O and the OH, that's the acid. Sure.
This whole thing's the acid group. Now, why is it an acid group?
You know,
Because the OH.
acidity.
Acids,
You're tripping on that.
acids are molecules that can donate hydrogen ions to in solution. So this hydrogen ion can come off of here, you see, and now fill the water with H pluses. And that's why it's an acid group. The H is not bonded too strongly, and it can come off.
Why is the other side the amino group?
NH2 is just called amino. I don't know where the name came from.
R. The R.
So we got a hydrogen sticking up here, and we got an R group. Now that R group, like you were saying, Chaz, it's variable. It can be one of like 20 different things. And that's why I have all the different colors here. Because this one has a different R group than that one, which has a different R group than that one. And actually, to properly show it, I would need 20 different colors. Yes?
Do the R groups make it like polar and non-polar and all that? Or?
Yes. So some R groups are what we call hydrophobic. Hydrophobic. What does that mean?
Water theory.
They don't like water. And that's all my green ones. All these green ones here are hydrophobic. They don't like water. And then all my red ones here are hydrophilic. They have hydrophilic R groups. You know what that means? They like water. So I'll stick some red ones here. Stick some red ones here. So if this was my whole protein, it would, if you dropped it in water, it wouldn't stay like a chain like this. What would it do?
The greens would go towards the end.
The greens want to get away from the water that's around them. So they would kind of huddle on the inside like this. And the reds want to go toward the water that's around them. So they would kind of stick out. So proteins, when you drop them in water, and you don't drop them in water, they're usually made inside a cell, which is full of water. Proteins won't stay just a straight line. They will fold into some shape. You all remember when we did the protein folding exercises?
I do.
Wasn't that enjoyable? I've got some other colors here. All the blue ones here have a negative charge. They're negatively charged. All the purple ones here have a positive charge. And so if you have a negative one in your structure and a positive one in your structure, what do you think will happen?
They'll attract each other.
They'll be flopping around, and they'll probably come together, and then they'll stick together like magnets. And that'll be your shape of your protein, with the greens on the inside, the reds on the outside, and those two stuck together. Isn't this cool?
Is that the order it goes in?
No, there's no order.
Oh, I thought there was an order. Didn't we do that?
Yeah, right?
Well, you got to worry about these first because they have the strongest
Yeah, that's what.
attraction. Well, actually, the one you have to worry about mostly is cysteine, which has a sulfur. And so cysteine, where's my cysteine? There's a cysteine right there. The cysteine amino acid. Let's go back. See the shape of the protein here. The cysteine amino acid has a sulfur hanging down off of it. And if there happens to be another cysteine, oh, there's another cysteine. Yay. If the two cysteines get near each other, they'll form a really strong bond that's really hard to break. It's called a disulfide bridge. And that'll stay stuck together. And that's really what holds the structure of a protein together. These disulfide bonds are probably the strongest bonds. And then these plus-minus magnetic interactions are probably the next strongest. And then the hydrophobic, hydrophilic actions are probably the weakest. So that's the order you're talking about, Josie.
Yeah.
Yeah. I probably should have shown it to you in that order, but it's fine.
Wait, are the disulfide ones are the strongest ones?
That's the strongest,
Okay.
the disulfide.
Sorry, I followed you.
So there's a lot of bonding that goes on when you're talking about amino acids. There's a lot of talk of bonding and structure and stuff like that. So, so here's the other thing about protein. We call this the primary structure of a protein. It's a long chain of amino acids. They're covalently bonded to one another. Do you know what we call bonds between amino acids? Peptide bonds. It's a bond between this nitrogen and this carbon on the next one over. So the OH will come off here, the H will come off here, and you'll get this peptide bond. You want me to draw it? OH comes off here, we form this bond, this peptide bond, and then you have your nitrogen here with a hydrogen and a carbon and a COOH. You didn't think I could draw an amino acid this fast, did you? Well, once again, you're wrong. There's your peptide bond. What's that?
Never doubted you.
Thank you. Thank you, Joe. Primary structure of a protein. Now, in water, it will start to form what's called secondary structures. It'll form hydrogen bonds between the hydrogens, these hydrogens here, or this hydrogen here, and maybe this oxygen. If you imagine this long string and it kind of winds down around here, you can have an oxygen attached to a hydrogen. Maybe it winds up like this. This oxygen attaches to a hydrogen on a different amino acid, and it'll form these hydrogen bonds, just like water has. And it'll make little curlicues. You know what we call that? It's a secondary structure.
Alpha helix.
That's an alpha helix. Or it'll form structures like this. Greta, you with me here?
I am.
What do we call that?
Pleated sheet.
A beta sheet or a pleated sheet.
Yeah.
I remember it. I did remember it.
So you have alpha helixes and you have pleated sheets. And that forms due to hydrogen bonds between the amino acids. They aren't as strong as the peptide bonds that hold the chain together, but they're pretty strong. What do you think? So that's secondary structure. Now what is tertiary structure?
When they like fold together again.
That's when that'll attach like that because of disulfide bonds. Or because of plus-minus interactions. And you'll get you'll get this part bonding to that part. That's tertiary structure. There's one more type of structure. You know what it's called?
Quaternary.
What's quaternary structure? That's when one chain will hook to a totally separate chain. Whoops. Now we have quaternary structure. Lots of proteins have, 4 chains that hook together to make the whole protein. We call this a subunit, and that's a subunit. And they hook together. Often it's called the alpha subunit, and the beta subunit will hook together to make the protein. Hemoglobin is a common protein they'll talk about on your test. It's got 4 subunits that all hook together to make hemoglobin. It's in your blood cells. A single blood cell holds about 1 billion hemoglobin molecules.
A single?
One blood cell holds 1 billion of these
So
proteins.
those are tiny.
And the blood cell is like the smallest cell in the body.
Mind-blowing.
1 drop of blood has like 5 million blood cells in it.
So that's a lot of hemoglobin.
Think of all the hemoglobin.
What would you do now? Put it
And you got more than 1 drop of blood in your body. That's a lot of hemoglobin. Why are you backing up? We're not done. We still got to do nucleic acids.
Heck yeah.
Alright, so nucleic acids are made of these things. What are they called?
Nucleotides.
Nucleotides.
Hey!
Adenine,
Sorry. You're upset.
There's 4 types, there's 3 things, in a nucleotide. There's a sugar, a phosphate, and a nitrogen base. Sugar, phosphate, nitrogen base. The nitrogen bases are of 4 types. Adenine, guanine, thymine, and cytosine. Now, RNA has uracil instead of thymine. The green is RNA. RNA doesn't have thymine. RNA has uracil. There's like 1 atom that's different between these 2. But it's enough to change the molecule. Now, G and A, guanine and adenine, are larger. Do you see how that's bigger than thymine and cytosine? See how it stretches out farther? It's actually a double-ringed structure. Whereas thymine and cytosine are single-ring bases. These are double-ring bases. And they have nitrogen in them. That's why they're called nitrogen bases. And so G and A are called what? Do y'all remember? G and A have a certain name. They're called purines. You know how I remember that? People from Georgia are pure.
And we have good roads.
That's right, we have good roads.
We're pure roads.
People from Connecticut are pyros.
Pyromania.
Pyrimidine.
My mom's
Pyro.
Or you could do Cleopatra and King Tut have pyramids.
Cleopatra and King Tut have pyramids. Retta, that is awesome.
pure graph.
So purines are the big ones, pyrimidines are the small ones, and guess what? They'll hook together to form a DNA ladder. So you can put, if you have nucleotides on one side, you can hook nucleotides to them on the other side. Click. Click. Remember this?
Yes.
Click. But it's always the case that if there's a purine on one side, there's a pyrimidine on the other. A always hooks to T, and G always hooks to C. Yes?
Isn't there like 3 to 5 chains going?
Yes, yes. There's a directionality, is what you're talking about. The phosphate end is the 5' end. You steal this C right here. So the 5' end is on the phosphate end. The other end is called the 3' end. So one side will go 5' to 3', the other side will go in the opposite direction. So we say that the DNA is in parallel, it's anti-parallel. This is another one of my dances at Ziggy's.
Do it, okay.
Anti-parallel.
Can you do the wave?
Pretty good.
Very good.
Wait, what's the 3' 5?
So the 3' is on the sugar end, the 5' is on the phosphate end.
Got it.
Wow.
That's great.
And then one last thing, ATP. Y'all remember the ATP molecule? Adenosine triphosphate. Well, it's got 3 phosphates instead of 1 phosphate. It's basically an adenine with 3 phosphates. And that's a common molecule used to carry energy in cells. Because the bond between that phosphate is easy to break. And when it breaks, it gives off energy, and it can store energy. You put in energy, and it forms the bond. So that's our energy carrying molecule in the cell. Woo! That was Unit 1. It's crazy. It's a lot of stuff. Can you handle all that?
Maybe.
Work on the problems and the FRQ. I'm actually going to give you a grade. You're going to get a 0 if you don't do these problems in FRQ. There's only 12 multiple choice, and there's 3 short FRQ.
Wait, are you grading on accuracy?
I don't know. I haven't decided if I'm grading on accuracy, but you at least need to do it.